Hi, my name is Mackenzie Hanlon, and today we're going to talk about positive inducible transcription. What is positive inducible transcription? This is defined when the regulator of the operon takes the role of an activator, meaning it is required to be in an active mode for transcription to occur, as well as having a stimulation from an inducer to begin a transcription. An example of a positive inducible transcription would be if you wanted to make a phone call, you need two things. You need a cell phone and service. The cell phone would be your activator, and the service would be your effector. This is because the phone allows you to make the phone call, but without the service, you can't get the call to go through. The phone call represents transcription. Positive control. This is when the activator or the regulator, also known as the activator protein, bonds to the DNA, which results in stimulation and transcription occurs. The activator genes are then expressed once the binding to the DNA occurs. Inducible control. The small molecule or the effector is present and binds to the regulatory protein, which results in the turning on of transcription. Then transcription will occur. How does it work? The activator is turned off or inactive until the activator becomes activated by the substrate that binds to it. The activator then binds to the promoter or the binding site, which is when transcription occurs. Histones. There are two types of histones. There's histones deacetylation, which is tightening the association between the DNA and the histones. This is used by an enzyme called histone deacetylase, or ADAC. Then there's histone acetylation, which loosens the association between the DNA and the histones by the neutralization of the positive charge of the histone. This enzyme is the histone acetyltransferase, which is also known as HAT or HAT. Keywords and terms. Our regulatory gene, or LACL, codes for the repressor. The promoter, or the LACP, binds RNA polymerase to allow transcription. Our operator, or LACO, interacts with the repressor. It's also known as the binding site for the repressor. Structural genes, or the LACZ, LACY, and LACA, translate and transcribe into proteins. Then you have your Z, Y, and A, and what they stand for, as you can see below. And then we have our polycistronic mRNA, which is translated into three different products right after being created by transcription. Our regulatory genes. This creates products that can interact with different sequences, which will affect the transcription and translation. Thank you for watching.